nowadays, if a night I would like to enjoy Hafiz Muhammad Abdullah on stage, like you can start a university.
First speaker of this session, writing like Dr. Muhammad Abdullah. sustainable of the landscape area on the shore of the
कौन सी वाली है? पहले वाली?
I'd like to thank you, understand the society, what is the science that are investing and activities of extent. And I ask us to provide intensity and by interest investing in us and invest. I also plan to present to move research paper that you have sustainable and big and true of the difference. The research exploration was based upon the following studies by professional experiences that invited living and science and literature. Introduction, the methods are considered government development, economic and educational study, and the wisdom about the state strategy. We find that they are criteria that must be consideration, considered water, preventive planning, question, safety, and research paper around access, the purpose of establishment of CPS is to see
इसके बाद कौन है? ये सारे तो ठीक है नहीं किधर गई USB में
Thank you, Dr. Rama. Is there any questions in the audience? We'll take the questions at the end. Thank you very
Yeah.
much.
सर
Now we invite the secretary.
अतर तारिक इनकी किधर है? आप हैं? अतर तारिक साहब जी इनकी कौन सी है? वो ऑनलाइन है या क्या चक्कर है? जी वो उनकी प्रेजेंटेशन कौन सी वाली है सर? अतर तारिक वो ऑनलाइन है, उनका नाम क्या है? अतर तारिक।
Our second speaker is Dr. Adhar from USC.
ATH आ रहा है ना, बट ये तो ऑनलाइन नहीं आ रहे। ये आ गया, ठीक है। इनको मैं करता हूँ, मैं इनको कह रहा हूँ अनम्यूट कर रहा हूँ। ये तो दो लोग अकाउंट से लॉगिन हुए हैं। कौन सी वाली? ये उनकी PDF है ना, PDF में शेयर कर रहा हूँ। उनको कहें ऑनलाइन आए और उनको कहें मैं अनम्यूट कर रहा हूँ, प्लीज अपने आप अनम्यूट करें। ओके सर, ये नाउ अनम्यूट और आप वो बात कर सकते हैं।
तस्वीर कहाँ है मेरी? प्रेजेंटेशन में नहीं है।
सर को बुलाएं, सर को नहीं फाइल डैमेज है।
जी अस्सलाम वालेकुम। हेलो
सर
आई
उनकी फाइल डैमेज
जी, मैं ऑनलाइन हूँ ना। हाँ जी, हाँ जी
PTT में डैमेज है।
ठीक है।
कोई और मैं कर रहा हूँ।
Hello, assalamu alaikum. This is Athar Tariq. Can you hear me?
Yes sir, we can hear you. You can move forward sir.
Okay. Bismillahirrahmanirrahim. Nahmaduhu wa nusalli ala rasulihil kareem. Rabbi shrah li sadri wa yassir li amri wahlul uqdatam min lisani yafqahu qawli. Dear conference participant, assalamu alaikum. I need to thank Qaid-e-Azam University, particularly Department of Plant Sciences, for inviting me to deliver a talk tonight. The topic of my talk is biodiversity of cucurbit crop and field detection of two viral and bacterial diseases. we need healthy food for nutrition. We get nutrition from our food, but we need to have some nutrition from healthy fruits and vegetables. And there is a recent book published, and the book name is Health Promoting Prospects of Fruits and Vegetables, and it has been edited by Leon Terry and published by Cappy. So I will be presenting my talk on cucurbits. Cucurbit is a huge family, and it is present all over the world. Cucurbit in Pakistan is represented by 95 genera and about 967 species. Mostly these are vines, and they produce some vegetable and some fruits for us. So most of the fruits and vegetables, they are pumpkins, squashes, cucumber, gourd, bitter gourd, melon, watermelon, cantaloupe, loofah, zucchini. And these are all vegetables, or these are all some of them are the fruits also, like melon, watermelon, and they are very delicious. So they have a lot of biodiversity, and in their size, in their phenology, and in their taste, in their color. This is the cucumber, and this is the watermelon. You can see that how, tasty that will be. And these are the cantaloupes and honeydew. The various sizes, various shapes, various color, and various tastes as well. So there's a lot of biodiversity. This is also another kind of cantaloupe, and this is all represent various cucurbits, and most of them are also found in Pakistan. And they are, as I mentioned, they are pumpkins, they are squashes, cucumbers, and you can see all of them. So if we have good crop, we need to have good seed and good agriculture practices, good farming practices as well. There is saying, you reap what you sow. Jaisa karoge, vaisa bharoge. So if you want to be a good seller, be a careful grower also, because there could be problem, and the problem could be multiplied, and it could cause loss of market or the additional testing. There are various diseases. So first we should have the healthy seed. We should have the good agricultural practices to get a good harvest, but we should also be careful with the diseases. So there are some commonly diseases found in cucurbit crops, as I mentioned earlier. So some of them are bacterial, some of them are fungal, and some of them are fungi and viral. And then there are some also nematodes and some other which cause the insects that also cause some of the diseases. So commonly found diseases of cucurbit are, I have mentioned, they are Phytophthora blight of pumpkin, powdery mildew, anthracnose, and white speck, downy mildew, pet rot, fusarium fruit rot, and leaf spot. And then recently there is another two other diseases found in California, and this is cucumber green mottle mosaic virus, or CGMMV. It is caused by some virus. And then there is other one found in California about 2 years ago, and it is known as bacterial leaf blight, or BLP. So I will be focusing my talk on these two new diseases. One is viral, and it is CGMVV, and other one is bacterial, or BLP. So this is how the symptoms are shown on melon, and the leaves are turned yellow, and the leaves veins are kind of also yellow, and leaves show cupping. And this is this viral disease of CGMMV in cucumber, and you can also see the symptoms over here. And these leaves, they show the cupping in them also, like over here. More prominent cupping over here, In also cucumber, you can see the leaves are cupped, and then they are also not very healthy, and there's some yellowish coloration in them also. This is a watermelon and how it is infected with CGMMB virus. It looks healthy, but it is disease. When you cut it open, you will find the symptoms. So when we cut it open, this is how it looked like. So the inside of the pulp is kind of rotten, or it is bitter in taste, or very bad in taste. The leaf are aborted, or they are not very much well developed. So this is the internal CGMMB symptoms in cucumber. This is more symptoms. I'm sorry, in watermelon, you can see the more symptoms in watermelon. So generally, these disease of viral disease CGMMB is found in watermelon, melon, cucumber, gourds, or gherkin. Gherkin is also kind of a cucumber, and it is used as a pickle. So we made pickle out of gherkin. So if the weeds are around the cucurbitt field or the plants, they can cause some diseases also, because there will be some insects staying over there, and those insects, they transmit the disease, and they transmit the virus, and the virus cause this disease CGMMB. And if there are some other solanaceous plant or chenopodious plant as a common weed around the field, they may also cause the problem. So we need to have a clear field where we are growing these cucurbitt plants. So how we inspect for this viral disease? So we need to inspect and collect any symptom possibly caused by the virus. We see the foliage, and foliage could show mosaic, mottling, distortion, vein clearing, or stunting. And if you see the fruit is mottled, discolored, distorted, and internal discoloration. And if we dig out the roots are reduced. So these are the symptoms of the CGMMB disease caused by virus. So if we have grafted cucumber or watermelon, they are more likely to be infected. And the cucumber leaf beetle, which is an insect or beetle, and the name is Rhapsodopalpa fascicolis, and this is a vector which may transmit the disease. The other one is bacterial flu diseases, and it is caused by a bacteria, and this is the symptom how it look like, and this is known as bacterial fruit blotch. So bacterial fruit blotch, or BFB, is caused by a bacteria known as Acidoborax avini, and subspecies Citroroi. So you can see the leaves are kind of infected, and they show stunted growth, and they show browning also on their surface. So this is another example of the picture of bacterial fruit blotch, and you can see how the leaves are infected by the bacteria, and they cause the discoloration of the leaf. So how we go for
slides share कर दिए, हमें slides नजर नहीं आ रही हैं आपकी, please. Can you share your slides, please?
Yeah, sure. You can see it now?
Not yet.
I'm trying.
Okay, if it's not possible, you can send us, we can share later on your slides. Thank you. You can continue your talk.
Yeah, it says the host disabled participant screen sharing. I have this on my screen. It says host disabled participant screen sharing. So someone over there on your end, they stop this screen sharing. That is why I can't share these slides. So can you allow me? Is that okay now?
Yes, we can see now.
Okay. It's clear? Should I go back or continue from here?
You can continue from the previous slides so that we can finish in time, please.
Okay,
Thank
sure. Yeah. So for bacterial, BFB, bacterial fruit blotch, collect the leaf with tan, dark brown, or reddish spots. It may look like anthracnose or gummy stem blight. Inspect closer at time with higher humidity and the temperature. So inspection time, when we should inspect. For the grower, as often as possible, look into any suspicious symptoms. And for the inspector or for the employees, we have two walks. First walk of the field is at bloom time or at early fruit formation. Second walk we can done at mature fruit time, that is every 6 row for bush squash or watermelon, and at further distance depending on the density of the crop. So this is how I mentioned earlier, the field should be clean. So clean field is necessary for the seed production because these two diseases are seed borne. So we should have clear field so that they don't have the insects or other viral or fungal diseases on the weeds. So fields should be clear of any weeds, stubbles, or any garbage or any stuff. If there are some weed may share some viruses and insect. So this is a field of a cucurbit, and you see how many grasses are growing around, and there are also some other kind of plants growing. So these plants and the weeds, they may host some insect or they may share some viruses and they can spread the disease. So we should have a clear field as I showed you earlier. There should be no weeds, no garbage, no stuff, no stubble, anyone in the field. So how we should collect the sample? Sample collection of sample at different ages with late and early symptoms. And we should keep the peduncle of the fruit with intact. And we need to separate sample with different symptoms, keep them cool, remove condensation, and then ship them cool and stable to the lab. And take the field picture, and take the record overview and the close-up of the field. So how we can manage the disease, these two diseases, CGMMB and BLB diseases, how we can manage them. First thing is removal of all viral reservoir. As I mentioned, there should not be any plants or any weeds which should harbor the insect and they are transmitted as a vector, the virus or the bacteria to the plant. We should keep the phytosanitary practices also, and we should do the crop rotation and cultural practices, and we should not grow the same plant each and every year. We should do the crop rotation. So then one crop is with the roots and root crops, and another crop is like other crops, fruiting crops. So we need to have crop rotation and cultural practices. So we need to have maintenance and management of moisture also. If there too much moisture, they invite more insects, more weeds, and they are also prone to the infection of diseases in the humid condition. But if there is drought, the disease is also progressing. So then we should use the certified disease-free seeds, and we should get this seed which are good quality and from a good firm or from a good company. And selection of a resistance cultivar another one to manage the diseases. And select the varieties that can be recommended for the area. This is most important because we should not do the same variety to all the areas. Their temperature in one area is different, the soil type is different, and then also day length and all other parameters, they are different. So select the varieties which are recommended for the particular area. Keep the field and the surrounding area free of weeds that harbor insects which can be spread viruses and bacterial diseases. And then we can remove plant debris from the garden after harvest, since many diseases survive on the plant debris and they stay from year to year. So this is a field worker ready to go to the field, and he should have a personal protected equipment and apron, cover his hands, and it has a long shoes and has Lysol as a disinfectant or some other chemical to disinfect his shoes and hands, everything, so they don't cross contaminate the field of the tools or the plants from one plant to other plant or one field to other field. So we should have our equipment and we should have the truck with water and tools so that we can keep the record. We should have the pen, pencil, copy, and the camera to take the pictures, and we should have all the things ready to go to the field and do the collection of these disease sample. And we should use the bleach as a disinfectant very safely. It should not split it here and there, and we should be very careful using the bleach. And we should also disinfect our tools, sterilize our tool with 50% ethanol or bleach, and also the tires of the and vehicles, so that when we go from one field to other field, we don't cross-contaminate the field. And also we should, after every collection, we should also disinfect or sterilize our tools. So thank you very much. So if we take the precaution, have good seed, and we have all the agronomical practices, we can have a good crop and we can have a good money, and we need to follow these instructions. Thank you very much.
Dr. Uzair is not here. So we move to next one, that is Dr. Azhar Rasool. His talk is tumor metabolism, novel and selective targets for cancer therapy. Dr. Azhar Rasool.
Assalamu alaikum. And firstly, thank you for the organizer for inviting me here. And please, if anyone can confirm me whether you can hear me clearly and you also can see the slides. So
yes,
today
we
I
can
am
see.
Okay, thank you. So as the the topic is tumor metabolism, the novel and selective target for cancer therapy. For the layman, what we can say that what we are planning or what you can understand to kill the cancer by taking away their favorite food. That's we are planning. And then actually I'm working in the GCUF, Government College University, Faisalabad, and I'm running the several projects. But today I'm going to talk only few of the snapshots of our one-up project that is about tumor metabolism modulators. So first, the question is tumor metabolism for cancer therapy, renewed hope or false promise. Actually, I think most of you are familiar with this word cancer. That's why I'm not going to talk about how this is a dangerous one and how many people are dying. If we just have a lighter note, even if we talk about in Pakistan, and only there are more than 200 type of cancers. But if we talk about a single one, just like a breast cancer, and we take the example of a single country, just like a Pakistan, so each eighth woman is diagnosing with the breast cancer in Pakistan. It means we all are familiar with this one, and this is in our surroundings. We know that someone is dying because of the cancer. Of course, it's a lethal one, and the people are approaching a different kind of approaches, including some are using radiotherapy and surgeries, and also other kind of the chemotherapy. But why we are focusing on the tumor metabolism? Similarly, some are using oncogenes and some transcription factors. So why the tumor metabolism? So firstly, if you look for that, and that portion particularly will be the more interest for the actually biochemists, and that is we majorly study in the biochemistry how tumor metabolism support the cancer progression. I just point out the few points. Actually, we know that we have a tumor, one cell changes and it is starting to grow up and becoming a big mass. So first, where the energy is coming up. So it means where the coming, that is actually metabolism. So first, what it do, it provide, assure the supply of building blocks for cell proliferation. Secondly, why that cells is growing, how the spaces are created. So what they do actually create the spaces by starving the neighbor cells. And similarly, it suppress the immune system, NADPH production, reduce the glutathione, and reduce the RAS by the mitochondria. This picture, I think most of the, of course, those people who are working in the field of biology, they must be familiar in the plant sciences or even in the animal sciences, and particularly the biochemists are much familiar with this type of diagram. And we know that, as you can see here, the glucose, there are more than 20 metabolic pathways are reported, including glycolysis, PPP. This is not a Pakistan People's Party. This is a pentose phosphate pathway. Similarly, the serine pathway. This is also called SBP, serine biosynthetic pathway, and the TCA cycle. Of course, I'm not mentioning all of these. Like I mentioned, more than 20, we have the metabolic pathways. And suddenly the people are working even on the single amino acid pathway, how that particular amino acid is formed. Like as you can see here, the serine pathway. In this pathway, we just focus how the serine is synthesized. And there are a certain type of cancers, actually, which depends only a single amino acid, that is the serine. So similarly, if we block that amino acid, particularly, we can stop that particular type of sub-cancers in which the serine is overexpressing actually. And that is actually the need of time as well. We can't treat every cancer by a single medicines. And we have to use a personalized medicine in that case. So if we talk about, if we give the simple definition of a single one, we know that the glucose actually, if we talk about the glycolysis, so most of the chemists already and biochemists know that conversion of glucose into pyruvate. Of course, this is a half definition, but we want to say that conversion of glucose into pyruvate with the help of different enzymatic reactions. And we know that there are 10 enzymes involved. But if we slightly go deeper, only in these 10 enzymes, they are not in 10 in number. Actually, they are 28 in number. If we talk about the last enzymes, as you can see here, the PK, that's a rate limiting enzyme. PK is again not an Amir Khan movie. PK here is actually pyruvate kinase. So that pyruvate kinase has a 4 isoforms, PKM1, PKM2, and then PKL and PKR. And more importantly, PKM2 is overexpressing in case of the cancer cells. Similarly, what I'm going to talk today specifically, I'm going to talk about this enzyme. As you can see, the ME and malic enzyme that we called it ME, and that's a malic enzyme. And this enzyme has a 3 isoforms, ME1, ME2, and ME3. So today I'm just going to talk what we have done so far regarding this enzyme and why we are focusing on this enzyme. And one thing I want to share here, that just for the interest and make you more clear, most of the enzymes, I don't know the reasons, but in Pakistan and other countries, we know that number 2 is always blaming, number 2 is not good. But that is also literally believe in cancer, mostly like I'm talking about PKM2, and similarly, malic enzyme 2, ME2 is involving in the cancer. ME1 is usually playing a role in the normal metabolism, but ME2 is overexpressed, then it is having a role in different type of cancer, as you can see here. Why we are focusing on that particular enzyme, that's a malic enzyme 2. So as you can see in this figure, this enzyme is overexpressed in number of cancers, including a lung cancer, prostate, leukemia, colorectal, and so on. This is actually our study. As you can see, Sarfraz and Rasool et al. We reported in 2018, and some other paper reported in Nature Chemical Biology, Nature Communication, and so on. But what we are going to with this enzyme, actually, there is a certain type of cancer where this enzyme is overexpressed. We only want to treat that particular group of cancers. But unfortunately, no potent and selective inhibitor of these enzymes are present. So therefore, not only for this enzyme, but for the other enzymes, my lab, we have a kind of this working scheme. Okay, we start with a molecular cloning. We start that particular gene and then we make it cloning, and then we have to express in the bacterial system, and we express that protein in our lab, and then we purify. And then I'm collaborating with one of the Japanese lab. We perform in that lab the chemical array screening, and we also have a natural product library and other by using an enzymatic assay. And then as you go through, we use the biological activities, flux analysis, and so on. But if you are interested, due to the time limitations, I know I have a 10 minutes, so I'm not going to see in detail. But if you're interested, this is a paper you can read out. This is also one of the collaboration from the USA, and also the author was also from the Quaid-e-Azam University. So we have this. This is also role in the glycolysis. So in this, you can see the detail further how we are working actually. This is our general strategies, how we are finding the metabolic reprogramming modulator. So screening strategies, primary screening, we are using a chemical array-based screening, and in silico, we are using a molecular docking in this one, and synthetic library in this we are also using. Then we use a secondary screening to reconfirm this one. And in this one, we have a natural product extract library, and fruit waste even we are using these days. Of course, number of the fruit we are using, but their waste products are, we are not utilizing actually. We are also just like that. This is libraries we have in the GCUF, and by using these enzymatic assay, we are screening against those enzymes. This is our general schemes. For example, if we take one of the extracts and we just assume we go for ethanolic extraction, then further steps HPLC, and then ultimately we believe in one compound more than we have so far 40,000 fractions, and we believe one fraction in our compound library might be 500 lakhs compounds. But this is just a brief intro. This is not into the interest of this one. But here we have the assay system that I'm collaborating with the Japanese lab. We have an array system. There's a number of papers as you can see here. If you're interested in these techniques, might be you undergo with these papers and you will see the detail of this technique. And as you can see here, these are all the enzymes working under different projects. As you can see, PKM2, ME2, 6-PGD. But as I mentioned earlier, these are the different my PhD students working on the one of each enzyme of the metabolism. And particularly today's my talk is focused on the ME2. So ME2, what we have done so far, firstly, after establishing the system for this enzyme, we have screened number of compounds against this enzyme. And out of those compounds, as you can see here, we have screened about 29,707 compounds. And after the initial screening, we find 196 hits. And then we using this assay system, as you can see here, this is a recombinant assay. We are, of course, this is a simple enzymologist, understanding easily. We are using a malate as a substrate and coenzyme NADP, and we are just measuring the NADPH endpoint. And if the NADPH level, and we are using our own purified recombinant malic enzyme too. So in that case, we reconfirmed, and out of these 196 sets, 2 compounds, as you can see, 8798 and 5734, they were more potent. And then further, of course, we find out whether these compounds have a drug-like structures or not. So we have chosen these compounds for the further studies, and we also consulted later on with the chemists as well. Okay, then we have synthesized, we are just, as you can see, the one of our lead compound that we given the name as 8798. Then we have done the SAR, structurality analysis, and we synthesize 100 compounds, and then we have 6 more potent hits, and we perform another technique that is called the SATSA. That is a cellular thermal shift assay. This is a more closer to actually the physics people. And in that, what we do, this is very interesting and simple technique. As we know that when we give a high temperature to any protein, it becomes degraded. But what we are doing in this principle, as you can see in this Western blot result, and this is a CBB to see it, we have loaded the same sample, but one sample where is our no ligand, that protein is degraded. And but where we have our ligand, our candidate, that protein becomes stabilized. As you can see here also in this one, one is we have a DMSO as a vehicle, and other we have our compound, target compound. Similarly, in this is more even clear. You see the band, then band is disappearing. Ultimately here you can see only band in our ligand sample, but there's a no band in the other one. And we have heated up to 70 degrees Celsius. Like as you can see, we started from the 45 and we start heating. So protein is degraded. That what we concluded from these results. So actually we concluded that our compound that we are sure is going to bind where this protein, and because of their binding, that protein becomes stabilized. So so far here, what we have we find that our compound inhibit the activity of malic enzyme and also bind somewhere. But exactly where it is binding, it is still we are investigating. Which residues is actually binding. We need other kind of crystallographic studies and so on. But that part we are still in progress. But later on, because I mentioned earlier, this enzyme has a role in cancer progression. And I want to mention another point. This enzyme has not only has a role in cancer progression, also particularly in the metastasis. So we are focusing, firstly, we check whether if this enzyme has a role in cancer progression, whether if we use an inhibitor of this enzyme, cancer growth can be limitized or not, can be inhibit the cell proliferation or not. But unluckily, as you can see, the first compound 8798 at 300, more than 300 micromolar, that was not cytotoxic. It means our target was not achieved. Similarly, we have another compound named as 8716, and but it has a 40 micromolar IC50, and we use it both ways. We also using with glucose and without glucose. We tried maybe only the glucose dependent, or maybe without glucose. We are focusing on the metabolism, so might be have a different activity. But there was also no major difference, and the IC50 was also very high. It's not so potent compound. So of course we got desperate. Maybe our purpose is not fulfilling. But I have another project, and my lab was also working, that was related to tumor stromal cell interaction modulator. In this method, what we were doing actually, we established one system. In this system, we have a cancer cell and we have a fibroblast and cancer cell. Actually, I give the common example. Of course, these days, again, you are listening, one of the person called Bhushan Singh, one of the Indian spy who entered in Pakistan. Similarly, actually, we believe, my groups and actually I believe on the simple science. And we using the same method, I believe, let's suppose the cancer, one thing I want to clarify here, if you stop the cancer at particular region, that cancer is not a lethal. Similarly, you have the cancer in breast, and if you limitize the cancer toward the only breast, 90% cancer is curable. Breast cancer is curable. But the problem, when the problem is started, actually the problem is started when cancer start to metastasize. When it's become migrate to other part of the body, that's the lethal. And then of course the doctor says, sorry, you are not going to treat it. So by using, but my question was that and many of you question is that, let's suppose you have a cancer in breast, but how the cancer migrate to the other part of the body, like how migrate to the lungs, how migrate to the bones, and how to migrate toward the brain and so on. Although this question is still a question, and I believe who will answer this question, he will be the Nobel right one day. But no one is know the question exactly why this is happening and why the cells are particularly moved to the particular organ. But what we believe, one thing other, we can say that, and according to the Kulbhushan Singh, why I quote the example, if Kulbhushan enter in Pakistan, he can't do any activity unless our local people not help him. Similarly, what I believe in case of cancer cell, when cancer start to moving to the different part of the body, unless the cancer cells, normal cells are not helping the cancer cell not do a lot of things. So by considering this simple science, what we did, we do the fibroblast alone. Fibroblast means the normal cells, and we grow cancer cell alone, and we do grow together. And we are very simple, who is working in the cancer biology, they know this assay, the migration assay, and that is also called the scratch assay. What we did, we grow the cells, as you can see in this animations, and when we just have needling by using only pipette needle, and we just give the scratch here, as you can see the scratch. By using this scratch, when we see the normal cell, the growth was slow. When we see the cancer cell, the growth was slow. But when we have a co-culture model, the growth was very fast, which indicate that there are a certain correlation between the normal and fibroblast. And actually, those fibroblasts which are helping here the cancer cell, they are actually what we call them CAF cell, cancer-associated fibroblasts. And that is one of my projects from the TWAS. I applied them. We want to see why they're collaborating, and we are looking this phenomenon actually what was the reason. And then other question is here now, if layman can ask, who is filling the gaps, whether the cancer are filling the gaps or normal cells are filling the gaps.
Wind up PGA, time
Give
we
a
have
study,
run out
and
of
with
time.
the green fluorescence and so on. In this, we confirmed that actually the cancer were not moving. The actually fibroblast cells were moving. And by using this assay, what we did, I put another grant and we asked them, we want to screen the library against this system. And we use a compound which slowed down the migration of the fibroblast when co-cultured by the cancer, but not in fibroblast alone at 24 hours. That will be our hits. But as I mentioned earlier, now I'm going to my previous study, ME2 inhibitor. And I told you earlier, ME2 has two roles, one in cancer progression and one in the cancer metastasis. And we just thought out that whether this compound can work in the cancer metastasis inhibition or in this co-culture model. And we were slightly lucky in this case. We found that NPD8798, that was non-cytotoxic. Keep in mind, ME2 inhibitor significantly slowed down the cancer cell-induced migration of fibroblast cells at 15 macromolar in co-culture, while has no effect on the migration of monoculture fibroblast cells. You can see with these pictures. These are the cancer cells. This is a control group. There you can fill up. And if you see in the NPD8791, they have slowed down the growth actually at 24 hours. So we assume that
Dr.
might be they're working. But
आप
still,
अपनी
yeah, I'm going to finish.
presentation
The last
कीजिए,
two slides. Last two slides. See, my time is going over, right? Sorry, I can't hear you. If my time is going to, I'm going to finish. I have just two last slides. Just in this one, so we believe this one, and then this is my PhD work. We have another hypothesis. Actually, if you look this picture, this is very interesting for many of you. We have developed the cancer model, and we are using a clinical drug. And as you can see, the tumor here, and of course, that when we use a clinical drug, the tumor has gone. But if you left that tumor for several days, and you have another picture, that's a cancer relapse. So now relapse here. So what we are planning, what these inhibitors, surgery, or if we have a chemotherapy, and after that, if we use this, our metabolic inhibitor has the potential to prolong this relapse. So what we are claiming, but still we need to clarify these points. And the next one, this system, this is also our proposed study. We want to check these compound on patient-derived xenograft model. We check on a view model. I didn't show the data here, but in the next phase, we are planning to take in the patient-derived xenograft model. If again, cancer biologists maybe understand well, here we will direct take the cell from the cancer patient, we will induce in the mice, and then we want to see. And by using that model, we want to check all these kind of parameters. I'm not going to detail as my time is going to finish. So in this systems, what we have done so far, so we have a established screening model. We have screened 10 targets like PKM2, ALDO, PGA1, but I have discussed only one ME2 with you guys, and identification and characterization of novel potent selective ME2 inhibitor. And why we are hopeful, this study could be the, it means in the clinicals or so on. So the reason is that, as you see in my last lines, phase 1 clinical IDH, there's another inhibitor that is also a metabolic enzyme, IDH, in one of the international company made and they have good safety profile. So we hope that our result will also successfully translate to the human tumor in future, but we are of course still need a lot of work. This is to achieve the impossible is to believe the possible. So this is my Chinese group where I done my PhD, my Japanese group where and this is my zoology university. And these are all the funders who are supporting me. HEC, recently I got also grant from the PHEC, Cisco, and TUAS and IFS, Biochemical Society, and most importantly, I would like to get 2 grants from the Nagai Foundation, Tokyo. So these are all the funders. And what I believe at the end, if you want to like the science more interesting, be interact just like a cell signaling pathway. So here are my collaborators from the different parts. So we are trying to collaborate with this one. So this is all about this 10 minutes talk. Thanks for your attention, and sorry if I was a little faster because of timing. And that's it. If you have any questions, I will be happy to have your questions. Thank you all, and thank you once again for the organizers for inviting me here.
Bismillahirrahmanirrahim. As our chief guest and guest of honor are here, so we would like to move back to our inaugural session, and then we will continue our technical session afterwards. So first, I would like to welcome our honorable chief guest and guest of honor and all other invitees that are present here. A very warm welcome and a very good morning from me and on the behalf of 4th International Conference on Biosciences. My name is Maha Munir. So before we start our conference, I would like to invite our chief guest, Professor Dr. Muhammad Ali, Vice Chancellor of Qaid-e-Azam University, to be seated on stage. Professor Dr. Muhammad Ali is the present Vice Chancellor of Qaid-e-Azam University, a very well-known personality, not only in academics or research based, but also famous among students. Now, I would next like to invite General Secretary, Professor Dr. Tasawwur Hayat, a very well-known mathematician. After that, I would present a warm welcome to Dean of Department of Biological Sciences, Ms. Safia. Next, I would like to invite ex-Vice Chancellor of Kohat University of Science and Technology, Dr. Zabta Khan Shinwari, to please come on stage. Now, I would like to invite Chairman Department Plant Sciences and Director Botanical Garden, Dr. Professor Dr. Mushtaq Ahmed. This international conference aims to bring together leading academic scientists, researchers, and research scholars to exchange and share their experience and research results on all aspects of biosciences. And I'm very thankful to the organizer of Department of Plant Sciences, Qaid-e-Azam University. And I'm very thankful to the organizers of Department of Plant Sciences and Biological Society of Pakistan that provided us with these opportunities with the facilitation of Pakistan Science Foundation and Pakistan Academy of Sciences. First, I would like to invite Professor Dr. Mushtaq Ahmed for an introductory speech on biosciences.
Professor Dr. Zabta Khan Shinwari, our professor in MSc and number of projects. And I'm very thankful to all of the organizers and especially the Pakistan Academy of Sciences and Biological Society of Pakistan, who gave us this opportunity to organize this 4th International Bioscience Conference. We got this target on 1st June and we just give a deadline of 1 week for the oral and poster presentation. So in 1 week, we got 1,400 oral presentations around the world. So that was a wonderful target for us. And we got number of the poster presentation around the world, especially USA, UK, Canada, Japan, Turkey, China, Malaysia, India, Ethiopia, Nigeria, so many countries have presented a number of the oral talks in this conference. So this conference is basically consists of number of the segments, that is the biological sciences, that is the botany, zoology, microbiology, animal sciences, pharmacy, biotechnology. So this is basically the conference about the biological sciences. I am very thankful to my respected seniors from Department of Plant Sciences and my faculty of Plant Sciences who help us and support us in this regard, and we have organized within a very short period of time. So the support we are provided by the Pakistan Academy of Sciences is immense important, and at the same time our Vice Chancellor support all of the ways. My respected Dean also support us very well, and we organize within a very short period of time. So hopefully you will enjoy this conference with a number of invited renowned scientists around the world. So this conference is not important for biological sciences, this is important for society, because biological sciences are basically the sciences which help us for the food security, energy security, and medicine security. In pandemic era, the biological sciences provide us a vaccine. So this conference will be very useful for the biological sciences, for the society of Pakistan and around the world. So this is a good opportunity for us to interchange with each other. We are looking that our department is a leading role in biological sciences in 2020 and 2021. The plant sciences is the topmost department in biological sciences, and we got a number of performance-based TTS awards 2020 and 2021. So we are looking forward that our department will be the top ranked department in Pakistan and in the world. So I'm very thankful to my respected teachers, my respected colleagues, especially the Director Student Affairs in charge and Director Academics and all the faculty members and students who participated in this conference. Thank you so much.
Thank you so much, sir, for your kind words. Next, I would like to invite Professor Dr. Zabta Khan Shinwari for a plenary talk on biosciences.
Thank you, madam. And I can push this back because I can. Bismillah ar-Rahman ar-Rahim. Very quickly. Worthy Vice Chancellor, Professor Dr. Muhammad Ali, Dean, Dr. Safia, Tasawwur Hayat, my friend Mushtaq, Tariq, Dr. Sarwat, and many professors and students. Assalamu alaikum. I will very quickly be, today my task was to just explain something. I'm very happy to see this biological society, sir. And I will be recommending that this society, we have to take it to international now. Nationally, I've been to two of your conferences. And those conferences, I'm really impressed the way the leadership, Professor Muhammad Ali is taking it on. Sir, we have International Association of Biologists, IFBA and many others. So I would recommend we link this to that. And how we link that today in 10 minutes, 44 slides, but 10 minutes. I'm just checking my time. Okay. Next, please. Who is good? You are here. I linked this, Dr. Muhammad Ali Shah Sahab, when they hired me as Professor Emeritus, one of the target was to talk to the younger student. And that younger, I taught a course last semester to BS on biodiversity. So I tried to link them to one health concept. Now, internationally, after this COVID, responsibility of scientists. Zinda rahenge to kuch karenge na. And for that, we need to do interdisciplinary work, social sciences and all other sciences, natural sciences. So if you look at this, this is the world map of extinction pattern. So far, Pakistan is, you look at the colors, the white means there is no extinction, yet we don't have data, in fact. But the other one, that on the top of the list, Hawaii and then South Africa, etc., etc. So there are a lot of problems with the plants, and then in turn we get diseases. In 10 minutes, I'll be trying to tell you how we get infectious disease, why we got corona, and what is it the first incident, or we had before also. So if you look at Hindu Kush Himalaya, the region that we are in, Pakistan is the only country in the world that stretches from 0 meter to 8,611 meters. No other country in the world. Nepal has Everest, but they don't have the beautiful Punjab and Sindh and Balochistan that you have. They don't have, they wish they could have a sea, a coast. You have it. So this is, God has gifted you the perfect piece of land. It's up to you scientists to explore it further. But the problem with this is, we are one of the Hindu Kush Himalayan main country. And if you look at the data, 25% of endemic species of the world are in Himalaya alone, and that are endangered because of various interventions that we are. And the importance of Himalaya, it feeds the glacier that we have, it feeds around 1.65 billion people, because the 10 rivers that are coming down, we have in Punjab 5 of them, but there are 10 rivers that comes from the Himalaya, and all of the agriculture and everything that depends on it. So indirectly, 3 billion people depends on this Hindu Kush Himalaya because of glaciers, because of water, because of biodiversity. So that's why you have to keep them really think. But what is happening? I will be touching about the modern technologies also, because I remain in biotechnology. Geoengineering debate in the world. I am a part of UNESCO, vice chair of the Ethics Commission. And there is that, what we are, the crazy scientists they are doing, they're thinking that we will pull all the carbon dioxide from the air, and we will try to, inject aerosol that can suck the carbon dioxide. But we don't know what Allah has done for us. If we interfere without having enough data, we will have problems like we have corona. So this geoengineering and that the ocean fertilization, there are two issues that we have to debate that will create a lot of problems for us. Jate jao beta quick. quick, मेरे हाथ को देखो जरा जल्दी करो बेटा ये time का. So when we talk about climate change, we usually say temperature and all this, but we rarely talk about the other issues that are related to climate change: civil unrest, commodity, food crops in future, what will happen to your food, etc. That we really need to debate. For example, if you have this problem of 1 degree centigrade high, your cornfield cannot, fertilize because of 1 degree temperature change in the area where you grow your maize crop. And same with sorghum, 25% will decrease if you have temperature, and all other cases that are related to this. Here, COVID is not the first pandemic in the world, and it's not that dangerous too. आपके लिए लग रहा है ना जो आप बंद हो, लेकिन so far we don't have that problem that we had in the history. There was once in North Africa and Middle East, 25% of the world population died because of that infectious disease that we had, and there are many others. I'm not going to talk but that was the result of what? Because we disturb the biodiversity and we disturb, vector, pathogen, and host, the ecosystem. And then you, as a human being, offer yourself as a very good host for the pathogen to come in and reside in you. So, the other thing, climate change, glaciers melt, and the recent study says that there's a very good preserve of microbes. For thousands and millions of years, they have microbes. And if they melt, you will get that as a gift. And this is what happened in Siberia. Anthrax were released, deer killed, and in turn it went, and there were a young boy that killed because of that anthrax. And we believe that 8 million old bacterium are in these glaciers that are being melted now because of climate change. And you will get a lot of pathogen if you don't, work on it. Thirdly, we have to think about invasive species. ये जो हम green Pakistan and all this imported species लगा रहे हैं, that is another issue that we have to really debate. You will have a lot of problems. South Africa had it. They spent billions of dollars in Cape Town because they introduced something which choked all the sewerage lines, and they were flooding. So that is what you need to think as a botanist, what to do with that. Because they drink a lot of water, they evaporate it, and you face a lot of problems. So, but is there any good thing with us, with human beings? Do we do something good also? Yes, of course. Because in the last decade, 2010, the maximum number of protected area in the history of the human being, we created, we preserved. So if the same discipline, if we go on, we can do a lot of good things, because if you do even in agriculture, sustainable agriculture, you can get from biodiversity a lot of fertile lands, etc. So the bottom line is that we have to work interdisciplinarily, because the reduced biodiversity, you will get a lot of invasiveness of the species. And that can from virus to the bacterium and to other pathogen parasites that you have that are linked with each other. You have to work on it if you really want to save yourself, humanity, as well as the ecosystem. Loss of one species, what we call it a keystone species, that has a lot of issues, that not only one species goes with it, the whole ecosystem is disturbed, and you have a lot of problem with your population. Jate jao beta. Data, I'm not going to explain it to you. The number of the species in the world, amphibian to other animals and plants that are being disturbed. And if one species, I talk about amphibian species, there are 1/3 of approximately 6,000 known species are threatened with extinction. And this one amphibian, here I show a marine snail. This gives you a kind of a drug that saves terrible cancer patients, what is called as zincotide. Antibiotic resistance, this is an ant that gives you next generation antibiotic. You remove this from the world, and you cannot resist the antibiotic. Now you feel the problem with this pandemic that we have. Thirdly, I will talk a little bit about synthetic biology, craziness of the scientist. We now are thinking that we can have synthetic dinosaur. We can have synthetic, so far we have synthetic polio. Without any template, we have polios from the chemicals. We have synthetic bacterium. We have synthetic other things. But if we do continue this, we will be facing problem in future. So we have to really think whether we need to have a dinosaur now or the other extinct animals now, really. That is what we internationally, we have to debate about the ethics of whatever we are doing. For example, the Spanish flu that killed millions of people just 100 years ago, 1918, we are trying to recreate it. We are trying in the name of vaccine, we want to have it again. And if it goes out of the lab, even in COVID, there are doubts that it went out of the lab. It's not natural. That we can debate someday again. But this is, if we do, we will create a lot of problems. Scorpion venom, the most expensive liquid in the world. That is, if you, that a lot of use of it. If in the next slide, you can see with the with curcumin and with the scorpion, you have a excellent drug that is being marketed in the world. If you remove the scorpion, you can face a lot of issues. Will we request for the taxonomy? Just one example. This was a plant that was discovered from Pakistan, a very good plant that gives a lot of good things for breast cancer. But the name that was given was wrong. So here is that, DNA barcode, taxonomy, and other modern disciplines. This was that paper. I was working with an American group. We finally told them that whatever you did was wrong. So the basic, you need plant taxonomy, herbarium, etc. And finally, little bit about the, the unfortunate thing in some of the selfish scientists are that we don't encourage interdisciplinarity. Now, this is the nanotechnology that we do from the plant. My group is doing it, and you get parts in the nano material that is really good that you can use. So you have to go out of you have to work with other discipline, and then you can finally get, again, theranostics and diagnostic through nanomaterial. Someday we can talk about it. Little bit for the leadership of the Quaid-e-Azam University. This is not only in Pakistan, but around the world. The universities have to change. The universities' disciplines have to change. They have to work with each other. They have this, I'm not talking about the Pakistani universities. Globally, we are debating the overall vision and mission of the universities to benefit the humankind. Not only this publication and all this, and there is that, in the global ecosystem, what needs to be done. And there is economies, natural sciences, social sciences, they have to sit together and design projects in such a way that they could really address the issues that they are there. And if you look at the next slide, go please. Around 50 years, this was a study, infectious disease incidents in the world, 335 infectious disease incidents, and all of them were somehow related to biodiversity loss. So that is that whatever you do, this is mainly you are responsible for the things that are happening in the world. Next, please. So there is that we really need to work. If we want, this Eid is not that Eid that you celebrate after a month, maybe again, or a month before. This is infectious disease, management that we have to work. And that is ecological, social, economics, and environment. We all have to work together if we want in future to avoid. Next, please. The problem with us is that in Pakistan, if you look at this map, the richest biodiversity area, the poor that they live there are the poorest. So we have to not only address the biodiversity conservation, but we have to address socio-economic development of those areas. Otherwise, simply saying, keep this plant that gives you a good oxygen is not enough for them. If they are hungry, they have to let down all this. One example of a federal, aage jato jo beta close karte isko. So we have to show solidarity with those poor people that they are there. The wealth creation only with few individuals is not enough. We have to, as our Islamic concept, we have to work with them. So one of the major conclusion of the recent study is of the social scientists that terrorism is not only because of religion. It is because of the socio-economic impact also. If the people are poor, they will sell their children and they will be involved in those. So it's to your benefit that you have to address those issues that they are facing. Jate ja beta. So finally, if we believe to conserve all this, we have to have a sort of environmentalism. This is a concept that we believe that we are running an uncontrolled experiment in the only house that we have. This one small globe, this earth. So we have to really, if we want to conserve, we have to work, and the peace, security, economic growth, and human rights, we have to address these issues if we want to conserve. Population growth is one of the important issues with the Pakistan that we have. And wherever there are population growth, there will be unrest, there will be governance issues. Pakistan is included in this. Look at Afghanistan, Niger, Congo, Pakistan. This is because of this population growth, one of the issues that you have to address if you want to have peace and harmony. And a lot of other things are related, but because of time, I'm just running up the slides. These are the hurdles, why we can't do it. Of course, bureaucratic, political, and other issues. The government has, in one of the slides, probably I'm showing that you have to involve biologists to take decision if you want to avoid these issues that they are there. The top 10, one is our food habit. The top 10 reasons, risk factors for the health is one of them is the low fruit and vegetable intake. The food habit, the burger culture that we have, this is international. You have to address this if we want to save people from the thing. So if we are scientists, we do research. Why we do research? Why science? We do this because we address questions. Questions for what? Getting answer right. And those answers for the humanity, those answers that benefit the humankind, not just to have science for the sake of science. So there is that you have to address. Lastly, this plant that you work on is not only plant. Like your gut has millions of bacteria, same as in plant. So you have to explore that, and that is where you can use those microbes for different kind of things. Look at here. These are the application of those microbes that we do. Our lab is working for industrial, for medical, for plant health, for other issues, and you have to address all this. So my message through the Vice Chancellor to the government is biology will play a key role in the future. And it has played, this is the century of biology. If you want to make humans safe, please consult biologists. They will show you how to work. And this is how policy making, just simply sitting in with economies and other will not work unless you address those issues that are confronted there. Thirdly, science literacy. I give you some of the example. Blood oxygen, they say, go and have a pressure cooker and you will get a very good oxygen there. So please, there are more safer ways to do it. So you need biology to teach right from the beginning, even to the social scientists, that how it works and what is important. There are a lot of examples that I have given here, but I'm just skipping those. And finally, infodemic, Ishtaq, the other way, talk about it. But the problem is that the scientists, they are hesitant or they are shy of the social media. You have to reach the community. You have to talk to them. And there is the research, action, and communication. Unless you have three of them, you will have these conspiracy theories. It happened because of G5. जब G4 आया तो ये हुआ, जब G3 आया, radio signal आया, 1916 में ये हुआ, they link all this. And you start believing it. Because you don't talk to them, you have no access to the society. So you have to have access to the society if you talk. Finally, we can reach up to the goal. The only thing is we need not a boss, but a leader. And you are a true leader, sir. Great, sir, doing all this. And that is the leader that I'm showing you on the slide. Boss super leke hurrah kar raha hai, but actually leaders are the people that learn from even these ants how to bridge when we are in difficulties, और कैसे अपने छोटों को आगे करके खुद उनको safety दे के आगे लेके जाते हैं. Biology tells you everything, sir. The only thing is that we have to. Thank you very much, sir, for doing all this.
Thank you so much, sir, for this piece of information that was very interesting. Before we move on to next guest, I would first like to introduce a new guest. Prince Naeem Malik, a very well-known businessman. Please give a round of applause. A very warm welcome, sir. Next, I would like to invite Professor Dr. Tasawwur Hayat, our guest of honor, to give us a piece of his thoughts.
Bismillahirrahmanirrahim. Professor Muhammad Ali, the worthy Vice Chancellor of the Qaeda University, Prince Muhammad Naeem Malik, great friends to several Qaedians, Professor Zabta Khan Shanwari, the ex-Vice Chancellor of the CUST, Professor Safia Saba, Dean Faculty of Biological Sciences, Professor Mushtaq Ahmed, present chairperson, ex-chairperson of the Department of Botany, Professor Dr. Tariq, Professor Sarwat, who is also along with us, the professor in the zoology, Professor Ishtiaq, several colleagues, ladies and gentlemen, distinguished guests, Assalamu alaikum. So it is, first of all, it is a great pleasure for me to be with you this time. And first of all, I would like to thank the organizing committee, especially the Professor Dr. Mushtaq Ahmed and his team, to invite me for this platform. Secondly, sir, I must be grateful to Professor Muhammad Ali, who spared time for all of us from his very busy schedule, along with the Dean Biological Faculty of Biological Sciences, several colleagues over here who spared their time for this event. Thank you very much, sir, and the madams for this. So what I want to do over here, just I would like to talk what is the role of this academy. The rest about the Department of Botany, I should say the Department of Botany now instead of the Plant Sciences, because I am highly scared from the worthy Vice Chancellor because he changed the name of the department in the last academic council, and that perhaps I am quite confident in the benefit of the faculty as well as the students, because what he told to us that at several occasions, especially in the FPC and other places, they were not going to recognize the name of the Department of Plant Sciences, and he proposed that perhaps Department of Botany is most beneficial for all of us. So thank you very much, sir, because in the history of the Qaeda University from 1967, this was first time proposed. Okay. Secondly, what should I say about the Department of Botany? I know about it very well. And I know because I am a pure Qaedian. And if you would like to ask from me that why I am calling as a pure Qaedian, because I came to this university in 1988, and in various capacities, I have seen this great institution, the Qaeda-e-Azam University, as a student and then as a teacher. So at various fora and in various capacities. So when we were the student, and even when I was appointed in the Qaeda-e-Azam University, at that time it was Department of Biological Sciences. And then when Professor Qasim Jan was the Vice Chancellor, in the end of the Dr. Ehsani Saab, he was the Vice Chancellor, then a proposal from the Department of Biological Sciences came that it should be converted into the Faculty of the Biological Sciences. And we believe that it was a very good idea. And now it is in terms of the School of the Biological Sciences containing various productive departments. No doubt several departments are contributing in the university through different dimensions. And in this history, what should I say that this department, either in terms of the department or the school, and a time was there when it was the institute before the department. So biology contributed a lot in the history of the Qaeda University and now contributing a lot. We are proud that several productive teachers are still there and they're contributing. And because of their contribution, the university is always getting further heights, either in terms of ranking or in administration or something like that. So no doubt in it, out of it, although there are several departments which are contributing, but Department of Botany is not lagging behind. They are contributing a lot over there. And Professor, including Professor Mushtaq, Zafar, Tariq, and several other colleagues also, they have a great share in ranking in the Quaid-e-Azam University. And it is pertinent to mention that this time also, Quaid-e-Azam University is now at 378th number in the QS world ranking, which is first time in the history of the Quaid-e-Azam University under the dynamic leadership of Professor Dr. Muhammad Ali, who is also contributing a lot because he is contributing in this university through different dimensions. Firstly, what should I say that perhaps in the history of the university, what I can say, because I am closely associated with this university from 1988. I have seen the pleasant environment in this university, which I have never seen before it. So, and the transparent policies and the academic vision is no doubt contributing, and that is one of the reasons that we are progressing day by day. And I am confident that, inshallah, that university in the next years would go at a better place, neither in ranking, but at every place. But I was asking from your chairperson, have you checked the ranking of the Department of Botany anywhere? But I could not get the answer. And in future, I should myself check, but I am quite confident that this department will have a prestigious place globally in the ranking if we would like to check it. And one thing that I want to keep you, that out of the another ranking, the Asian World Ranking is also announced, and Quaid-e-Azam University is at a very high place in Asia. So this is also good achievement over there. Thirdly, which I would like to mention over here, that according to the QS-based criteria, there are 5 parameters for the evaluation for it. And you would be surprised to know, although the Vice Chancellor of this university knows, and several other colleagues know, but perhaps the students does not know, that I would like to mention that there is a parameter about citations. And Quaid-e-Azam University for the citations evaluation score is 98% out of 100. And that is quite far more. And I was checking yesterday, even from MIT, Stanford, and Caltech too. So this is a good sign that what we are doing, although we need to do a lot, this is not the only way that we have to stop the things, but I mean to say is that the encouragement that we are receiving from the administration and the high-ups nowadays, I hope that with the continuation for that, the university would be sooner. We are expecting that that should be in the next years would be around 250 or 300, and that should be our target. So this is all about the department. Now I should come back about the everybody know that up till now, the Quaid-e-Azam University is maintaining its flag to be the best university in Pakistan. That I should say in this one. About the Pakistan Academy of Sciences. The Pakistan Academy of Sciences was established in 1953, soon after the independence of Pakistan. So it was established through the vision of a few distinguished scientists. In the dictionary of the Pakistan Academy of Sciences, we are calling them the founding fellows. So they sit together, and at that time, they proposed the idea. And according to their vision, this academy was established in 1953. But interestingly, what I want to mention over here too, that from that era, even in the establishment of the Pakistan Academy of Sciences, there is a great role of the Quaid-e-Azam University, because the Professor Riazuddin Siddiqui, who was the Vice Chancellor of that university, Professor Riazuddin, who was at that time the director of the Institute of Physics, and that kind of the people were involved. in the establishment of the Quaid University. And I mean to say that when I was listing, out of the 9 founding fellows, there were 3 founding fellows from the Quaid University. So there is a great share even of the Quaid University in the establishment of this academy from the day first. So that is also a great contribution out of nation. And secondly, what I would like to mention, because that is also quite important, otherwise Professor Zapta would not chase me out. So there are up to 11 Secretary General of the Pakistan Academy of Sciences from 1953 up till now. And out of these 4 are from the Quaid University. And Quaid University was also headed as a president Pakistan Academy of Sciences also. And along with that, the present Vice Chancellor of the Quaid University, Professor Muhammad Ali, last year is also declared as the fellow of the Pakistan Academy of Sciences. So I hope that, sir, we would inject to you quite soon. So, and this academy would be benefited by your great vision and dynamic personality. So that is also. At present, Pakistan Academy of Sciences has 81 fellows, 33 foreign fellows, and 23 members. And when I was looking the list of the members, so out of the 23, there are 11 members from the Quaid. When we would like to talk about the gold medals or something like that, then just 2 years back, although we are taking our share through stringent criteria approved by the general body for the evaluation of the gold medals or something like that. And every year, Quaid University is taking its share very well. And almost 2-3 years back when we were looking, so Quaid University was taking about 50% share from this academy. So that is why how Quaid University is playing its role in the development and in the success. This is the success story of the Quaid University and what Quaid University is having a role nationally and specifically in the Pakistan Academy of Sciences. So what this academy does. Pakistan Academy of Sciences basically provides a vision to the government for the policy documents, something like that. And we are taking lead into it, that already what was done and what we want to do now. Now we are focusing that the high-ups in the government must realize that what is the importance of the Pakistan Academy of Sciences. And basically the scientists are those basically that can realize them that without the scientists, which are in the form of a pool, in the form of the scientific talent in the country, they can bring the change in this country. And that is quite important. And for that, we have established a policy unit, which was not in the past. And through the establishment of the policy unit, I am confident that Pakistan Academy of Sciences will do now in this direction, that how the government can be connected with the scientists through this fora. And already Ministry of Science and Technology, Ministry for Climate Change, Petroleum, and the Ministry of Education are working with the coordination of the past through different policy documents. Another activity, including the 2-day activity, for example, the seminars, conferences that are going to be sponsored by the humble contribution through the Pakistan Academy of Sciences. Already, I have received the order of Professor Zapta Khan Shahwari and Professor Mushtaq Ahmad when I entered today this morning, that this academy should sponsor this event too. So, sir, we are ready for it. Academy is on your disposal. So whatever you would like to expect from us, we are on your order, sir. Just place it. And I am also biased towards the Quaid-e-Azam University. That is not, but I am quite confident that any activity nationally, we always sponsor. The academy is not so poor place, although it was in certain time, but no, I hope that we can manage the things, but the members, fellows must move forward for the activities which are useful to our young generations and something like that, because I believe that no young minds move forward without the encouragement and active support from the patronage institution, and Academy of Sciences is there to have a role for it. So this is all that I want to share. And now, what Pakistan Academy of Sciences contributed for the institutional development. Up till now, I would like to say that Pakistan Institution, Academy of Science has developed one center in NCP, National Center for Physics, another in Zabest, not Zabest, there is something like that. There is another private Nano Center through the Preston University. And third, which I would like to say, which is a huge mega project, Pakistan Academy of Sciences contributed into it. And that is the Chanapak Giant Research Center of Earth Sciences at Quaid-e-Azam University. And I am quite confident that would change the face of the Quaid-e-Azam University under the dynamic leadership and great vision of Professor Mohammed. Because let me own this, that he is the only person, although Academy of Sciences started that center, proposed that center, worked behind it, but that was the idea through joint collaboration with the Chinese government and the Pakistan government, because I should not be biased, but he snatched that center from another institution to the Quaid-e-Azam University. So, sir, thank you very much, because I know that how much powerful the other institutions were to take it, but ultimately we are successful in getting it out for the Quaid, to house it at the Quaid-e-Azam University. So that credit goes to you, sir. And I am confident that we will not disappoint you, sir, with your active support, great vision, and promptness. I am realized that government is looking at the university in a prestigious manner at every forum, neither in HEC nor in ministry, but everywhere. And this is also a good sign for the Quaid-e-Azam University, because there was a time unfortunate when the past glories of the Quaid-e-Azam University were lost, and ultimate through you we revived those, and that is also a good for all of us. So at present, Academy of Sciences, in addition to all these things, is also publishing 2 journals already, but we have decided to start a new journal in the engineering sciences and emerging technology. One journal in the medical sciences, we are going to plan to start it very soon. And another, maybe later, we would like to start in the social sciences. So we are trying to enhance certain activities in the academic dimensions too. So last but not the least, I am thankful to all of you for your sincere efforts in organizing this event. And sure, credit goes to all the speakers, either they are present physically or virtually. And this Academy of Science, I am again repeating, is on your disposal. Whatever you people need from here, just write lines, and I am confident that we will try our best to manage the things over there. Along with that, thank you very much, sir, and for your time and efforts. So may Allah bless you all. Allah Hafiz.
Thank you so much, sir, for your kind words. Now I would like to request Professor Dr. Muhammad Ali, our chief guest, for an inaugural address.
Miss Safia Ahmed, it's so good to listen that she's gone back to being very young, and all our faculty of the Department of Botany, students must be thinking that why we have renamed it as a Department of Botany for the many reasons that our students were facing a lot of problems while being recruited through public service commissions. And that was the reason, because certainly plant sciences are more catchy name, but we wanted that we have to look at the employability of our students. And botany is again a beautiful name, because we talk about botanical gardens and botany and the environment. And so Professor Mushtaq, Professor Tariq, Zafar, and all the faculty of Department of Botany, Dr. Sarwar, welcome back on campus. And Dr. Zabta Khan Shinwari he's an ex-vice chancellor of Kohart University of Science and Technology, but currently is a professor emeritus at Qaeda University. Naeem Malik is my old friend. He's an international citizen who's basically has a business interest in the United States and United Kingdom, but he's a frequent visitor to Pakistan. And now he has many friends at Qaeda-e-Azam University as well. So he's a friend of Pakistan as well as friend of Qaeda-e-Azam University. And all my audience who have joined us virtually, this is indeed a great pleasure that I am part of this ceremony. I would like to apologize that I was a little late for this inaugural ceremony, because I was in the BISP board, and where we had to make a decision to get almost 10 billion rupees more for the needy scholarship students. And we were very successful to convince the BISP board to allocate additional 10 billion rupees for. So that was the reason that I was late, because I was trying to argue my point that this is a very big and productive investment on part of BISP and SAS, that if you sponsor a scholarship, it is a real poverty alleviation through empowering the needy students. So this is the reason that I was late a little. But looking at your different presentations, there was another very interesting thing, that Dr. Azhar actually presented a paper to which this is NRPU project. He's my student, and I am co-PI in that. So I just sent him a message that you just forgot my name, and it was more, presenting the name of the Chinese. It's good, Chinese are good friends. But I have a lot of interest in, I'm basically a man from the zoology, but I've done a lot of work on medicinal plants, even using different plant products for natural coloring of our fabrics, because I also like fashion, and so I did some work in how to use these plants as a beauty for beauty products, and for giving a natural dyes to our fabrics, so that the dyes are less harmful. How to produce even a colored cotton as well. So there are many things which very, very interesting.